Laying the groundwork for a new discipline requires the coordinated activity of educators, scientists, engineers, and the commitment of large organizations and universities. In establishing the Center for Engineering Mechanobiology, we have assembled the very best in our field from multiple institutions to build upon our strengths to establish a new center. This center will change the future of biomedical research and develop new technologies based on mechanobiology. The Center for Engineering Mechanobiology represents the culmination of efforts by scores of investigators and institutional leadership from the University of Pennsylvania, Washington University in St. Louis, Alabama State University, Boston University, Bryn Mawr College, and the New Jersey Institute of Technology. Together, we have put together a center like no other. Our team is committed to advancing the field of mechanobiology through novel research partnerships, innovative educational practices, and outreach activities focused on growing our community and training the next generation of mechanobiologists. Our center, by its very definition, is a multidisciplinary one. Our scientists are partnering across scales and across kingdoms, from plant to animal, to develop new insights that will move the field forward to develop the new knowledge that will spawn the novel technologies of tomorrow. The University of Pennsylvania will serve as a central focal point of the new center. Penn offers a compact urban campus featuring a wide number of research centers that interact and collaborate, finding innovative ways to bring disciplines together. With all of Penn's professional schools and graduate groups located on one campus, innovation and interaction across disciplines comes naturally. The same is true at Washington University, our principal partner, where top-ranked engineering and medical schools are located in close proximity to one another. Additionally, our partners at the New Jersey Institute of Technology, Bryn Mawr College, and Alabama State University are leading the way in developing advanced biomaterials, studying cell-tissue interactions, innovating in education and outreach. For a new center such as this to thrive and grow, we must be at the forefront of developing new technologies to rapidly expand this new field. For this, the Center for Engineering Mechanobiology is taking advantage of the rich resources available amongst its center members. At Penn, beyond the innovative work going on in the labs of center members, we have unique resources that will help us to move our science forward and train the next generation of mechanobiologists. For instance, Andrea Stout runs a microscopy core.
I'm Andrea Stout, the director of the CDB Microscopy Core. We're in the Perelman School of Medicine here at the University of Pennsylvania, and although we largely serve investigators at the School of Medicine, we're the main microscopy core for the entire Penn campus, including other departments and other schools like the dental school, the vet school, the engineering school, the Center for Engineering and Mechanobiology. Many investigators come to use our confocals because they don't necessarily have access to high-end equipment in their own departments.
In addition to this more traditional imaging that we can do in the microscopy core at Penn, we can also take advantage of imaging resources that let us visualize things on the nanoscale.
My name is Dwight Williams. I'm the director of the Electron Microscopy Resource Laboratory at the University of Pennsylvania School of Medicine. We specialize in electron imaging, both SEM and TEM, so we're concerned with things in the nanometer range of imaging and below. This microscope behind me is the TF20, our new microscope, which has a direct electron detector for doing single molecule data collection so that we can do single particle image averaging and protein structure determination. An example of some of the work that we do at the Center for Engineering Mechanobiology is here we're imaging microtubules where they're bound to dining in small nanorods, which are fluorescent in the light microscope. To understand their orientation with the microtubules and the dining, we image these in the electron microscope.
In some cases, commercial technology is simply not enough to see what we'd like to see. In that case, we build our own unique imaging systems.
Hi, I'm Deborah Schroeder. I'm a graduate student in the laboratory of Yale Goldman and the Department of Biochemistry and Molecular Biophysics. I am currently studying the mechanobiology of two molecular motors, myosins 5 and 11. So I'm going to be showing you the principles of polarized turf microscopy. And what we use this for is to look at single molecules and look at the orientation of small parts of those single molecules as they process through time. So as they're doing their thing, we're looking at one part of the molecule and seeing how it moves. So we've used this for a few different types of small molecules so far. We've looked at the lever arms of myosin, which is a molecular motor, and we've noticed that as it walks, a single molecule has lever arms that actually change their orientation as they are walking along.
Sometimes we also need to see things very quickly in multiple channels to monitor and view rapid interactions between molecules. For this, we turn to DiscoTurf.
Hi, I'm Michael Woody. I'm a fourth-year biochemistry and molecular biophysics student in the Pennsylvania Muscle Institute in the physiology department. And I study the mechanobiology of motor proteins using this advanced optical trapping and fluorescence imaging setup. And so we have four-color fluorescence imaging that we can do turf, and in fact, a type of turf we call DiscoTurf, shown here, where we can do single molecule experiments at a very high resolution and eliminate any fringes that would come up in typical turf experiments. So we can do single molecule fluorescence experiments here, but we also have an optical trapping setup, which we've home built to do high-speed optical trapping using a sophisticated force feedback system so that we can see very short interactions between two molecules that can either produce force or sustain force, and we can get an idea about how those molecules interact with a time resolution on the order of nanometers, piconewtons, and microseconds. So we have very high time resolution that we can apply in multiple ways here to study molecules that both produce force and can move things around inside of cells.
Probing the biomechanics of both native and engineered systems is an important component of our work in mechanobiology. Our center has rich resources that enable us to measure mechanical properties from bulk tissues down to the micro and nano scales. In particular, the NSF-supported NanoBioProbe facility at Penn is a unique lab that serves as an incubator for new probes of nanostructure. This facility is equipped with multiple advanced scanning probe systems, including 2 atomic force microscopes regularly used by center members. A center such as ours needs ample computer power to solve the complex models of cell behavior that will be developed. Resources are available across the consortium for this purpose. For example, the Penn Institute for Computational Science is a cross-disciplinary institute for the advancement, integration, and support of research via the tools and techniques of high-performance computing. Their Walnut Street high-performance computing cluster is a shared facility that can be used by all center members. Indeed, the primary workload of this cluster is computationally intensive problems covering fields such as soft matter physics, material science, and mechanical engineering, all relevant to our new center. Microfabrication is an important tool that will be used by all center members across IRTs and working groups to advance our understanding of mechanobiology. Resources abound for this at each member institution. For instance, the Singh Center for Nanotechnology houses an extensive array of facilities and equipment for nanofabrication. This includes specialized areas for soft lithography, an important resource used in the construction of our organ and plant-on-a-chip devices. Bryn Mawr College, located just 15 minutes from Philadelphia, also has excellent nanofabrication resources. For instance, the Nanomaterials and Spintronics Laboratory has a sputter deposition system and mask aligner. These state-of-the-art tools enable fabrication of a variety of single or multi-layered thin film samples or pattern devices with sub-nanometer thickness. These facilities have been used by both Bryn Mawr and UPenn researchers and will be open to other institutes in our center. Additionally, at NJIT, there exist very high-quality, industry-standard Class 10 cleanroom facilities for nanofabrication. This center-wide resource will provide the highest quality microdevices to center members. Importantly, the National Science Foundation recently supported, for 7 consecutive years, the NJIT Summer BioMEMS Institute that utilized this facility. Indeed, the lessons we learned from this activity will be incorporated into our educational boot camps for new center members. One of the interesting features of our new center is its desire to cross kingdoms, from the plant to the animal kingdom. Specifically, investigators at Washington University are at the forefront of studying how physical forces influence plant growth and development. This includes specialized laboratories where individual plants can be exposed to physical features from their environment and their growth can be studied. In addition to their expertise in automatic phenotyping of how physical perturbations alter plant growth, center members at Washington University also have advanced tools for studying plant composition across length scales and at all points of their growth cycle. For example, The high-resolution NMR facility in the Department of Chemistry is used for protein structural and dynamic analysis, as well as for plant cell wall biochemical composition and architectural analysis. This includes state-of-the-art field emission SEM, secondary ion mass spectrometry, and X-ray photoelectron spectroscopy systems. Biomaterials are a central focus of IRT3. Investigators throughout the center are developing novel materials and fabrication systems to engineer in complexity in 3D structures. With teaching and learning at the core of its mission, the Center for Engineering Mechanobiology is at the forefront of using creative approaches to education. Taking advantage of new active learning facilities, like these located in the Penn School of Engineering and Applied Science, and across the consortium, we will organize a robust educational program to train the next generation of mechanobiologists. As we sat down to think as a group about forming the Center for Mechanobiology, we started from very different places. We developed a shared vision. That shared vision spans length scales, and spans time scales, and even spans kingdoms. We're committed to working together to develop new knowledge to train the next generation of mechanobiologists, and ultimately to transform the field of mechanobiology.